Hello, all. I'm going to try to get this. It may be a two-parter, but the recording for the review for our exam. And again, I just look at the exam and sort of narrate over it. And, and I did take a kind of a look at it. I think there were 70. I just have to open it again, and I will let you know. Here it comes. Seems pretty juicy for a first exam. Yeah, 70 questions. Lucky people. All right. So again, it's all, Scantron, number two pencils. We've been this. What we're going to be covering tomorrow, today I'm doing this today on the 6th, tomorrow will be the 7th, depending on when you listen to it. We're going to talk quite a bit about the neurotransmitters. And that's really the thrust of all this. I just got started with a little bit about acetylcholine. But understand, basically, the way the question will set up, there'll be a key, and heck, I'll tell you the key: tyrosine, indolamines, catecholamines, gaseous agents, and enkephalins. That's the answer key: A, B, C, D, and E. So, as you can guess, there's going to be a series of questions associated with it. And basically, the way it starts, where they fall, which would be, an endogenous morphinic compound, which would be have both neurotransmitter and endocrine applications. Those types of things. I think that's kind of where you would look to answer those questions. Which one's a biogenic amine? Which one is responsible for the production of epinephrine and norepinephrine and dopamine, et cetera, et cetera. So those are all kinds of things that we haven't touched very much yet in the role of gaseous agents, but this is a semester we spend a lot of time with this, and so I think you would probably find it uh, fairly interesting uh, when we do go over it. But that's the nature of the type of questions. So rather than say, "Oh, tyrosine makes dopamine, norepinephrine, and epinephrine," then the second level is what are they used for? So dopamine, we'll see plays an intimate role, and we've already discussed it, but it certainly plays an intimate role in Parkinson's disease, as produced in the midbrain. We'll take a quick look at that again, as is norepinephrine, but epinephrine, uh, very very similar structurally to norepinephrine, happens to be produced in the adrenal gland. So it's a double duty type of thing. So it plays a role in, as you can tell, nerve and endocrine. That's the nature of those types of questions. So we'll start there. We look Many of these work on that G protein linked pathway. Okay, with primary messengers and that type of thing. So most of them work externally and bond themselves to some type of G protein and sets off that repetitive pathway we've seen so many different times. The roles of cyclic AMP, the role of adenylate cyclase, the role of GTP and GDP. What are the second messengers? Down the road, we'll see the role of tyrosine kinase, though we don't necessarily see it here. So, what are they associated with? What do they necessarily do? That's a question that's in there. Okay. So, all of those are areas. So, we begin to look a little bit more in detail in that. I would think this would overlap relatively neatly with cell and molecular. I don't think there would be a particular problem with that. It's there. So exactly where it works. Remember, when we're utilizing steroidal based or fatty based receptors, they have the remarkable ability to transit through the membrane and basically bond with intracellular receptors within the cytoplasm, and then that unique combination is able to then effectively make its way into the nucleus in the eukaryotic cell, our cell, and subsequently initiate transcription and translation. So it's a totally different mechanism than that chain reaction we associated with frequently, let's say, with those G protein based reactions that are there. So I think that's an important distinction to be able to look at. So we were going to look at those much more closely. Again, you're going to see when we get particularly into endocrine, you're going to see the oddball there. Even, and it also is made out of tyrosine, it's thyroid hormone. Thyroid hormone is a very interesting agent because it basically takes, if you want to think of it this way, an upside down and a right side up tyrosine, very symmetrical, and adds either 3 or 4 iodines to produce a very interesting molecule that's amino acid based, yet the way it acts is as a fat soluble base, as a nonpolar covalent. It acts like a cholesterol. It works the same way. It's an intracellular rather than extracellular receptor because of its relatively diminutive size and the symmetry. And remember, when it comes to polar versus nonpolar covalent, it's all about symmetry. You've had the cell cycle ad nauseum, you're going to have it again. Where do things occur? What occurs in G1? What occurs in S? What occurs in G2? What are those basically channels that are involved that control it? Those stop signs that are there that relate to the production of cyclins and the bonding with cyclin depend, dependent kinases. Why are they there? What happens if those conditions are not met, etc. Those are the kinds of questions you will look at. So in the cell cycle, where G1 is all about growth and replicating macromolecules and basically assembling all the ingredients necessary not just for duplicating the cell, but also for duplicating the genetic material. In S phase, it's all about duplicating the DNA. In G2, it's assessing that everything is ready and effectively intracellular mechanisms assessing, molecularly speaking, that all conditions are met to proceed into mitosis. And that's the purpose of those. G1, you can't pass through that control center unless you've met certain conditions producing the cyclines. And once again, there'll be, again, cyclin dependent kinases allowing you. And we used to separate the G2 and M, but they're really now, they basically function. If you pass one, it goes directly to mitosis. Obviously, all the events of mitosis are important. What happens? Okay. How do we prevent it? What's the role of apoptosis? When do cells go into G sub zero? When do they effectively exit mitosis and exit the cycle? So, all of that type of stuff that you have to know. Again, I emphasize that biological systems inherently are prone to acceleration. We tend to do things too rapidly internally. Our heart rate is governed, our cell cycle is governed. When we lose that governance, things begin to spin out of control. And nothing is truer than the cell cycle. What happens when you do anything too rapidly? You make more mistakes. Eventually, you can't compensate for those mistakes, and mistakes become really problematic and particularly sometimes devastating. Effectively, cancer is the end of that tree when you get to the point where now you can't even recognize what you were starting with, and it has properties that are inherently destructive effectively. And that's what it is. It's mitosis gone out of control. And then we lose all of those safeguards and all those wonderful checkpoints, and there you go. So again, I like to look at things globally like that and understand it. And you'll see when we get into the autonomic nervous system, roll the vagus nerve, majorly to control your heart rate. The healthier you are, the trimmer you are, the more you exercise, your resting heart rate's a wonderful indicator about that. We go into that chapter and verse when we do the cardiac unit, which I always find to be very, very interesting. So what do they particularly do? Okay. Now, the 4 events of prophase, without going into the early or late prophase, I'm just not that interested in it. The dissolution of the nuclear membrane. Again, it's a membrane. It can separate and it come back together just like it does in exocytosis or endocytosis. So you have the nuclear membrane effectively opens up so we can't really visualize it. The nucleolus begins to disappear. Same logic. It will reform, whether there's one or more. Okay. The change in the chromosome structure, really the most significant event. The development of the supercoiling, and certainly the start of the microtubular structures that eventually become the spindle, all reversed in telophase. Metaphase, they line up on the equator, the so-called metaphase plate. Anaphase, shortest of the ones, they pull them across. separating the sister chromatids, and voila, next thing we know, we've got the cleavage furrow starting to form. We've been into full-blown telophase, reverse prophase, and subsequently cytokinesis. Mitosis is nuclear division. Cytokinesis is cellular division. I can't tell you how many AP1 students blow that question. What's mitosis? The cell divides. True or false? False. Oh, you're probably nuts. Okay. About mitochondria, we know the uniqueness of mitochondria in our cells, that they have the endosymbiotic theory basis, and they have prokaryotic DNA, prokaryotic ribosomes. They are uniquely structured in such a way that it clearly indicates it's been an absorbed prokaryotic organism that has become one of the foundations for developing membrane-bound organelles. We can see the similarities that are there. It's fascinating in all its phases. So when we exercise, when we want to make more, we make more. If we slow down, we make less, because they certainly have a lifespan. So all of those types of stuff. Exactly. So it mimics it's almost like a prokaryotic cell inside you. It mimics that activity. Be familiar with its definitions. Is it membrane bound? The two membranes, how they work, all the folds for surface area, blah, 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 blah. Peroxisomes and lysosomes. Lysosomes are very specific. The lysosomal enzymes, acidically based, function well in lower pH or with the aid of protons, are particularly for digestion and defense. Where peroxisomes, I mean, they certainly have some role in probably lipid metabolism, but more so than anything else, it's those toxic derivatives of oxygen. It's changing the superoxide dismutase mostly, and other derivatives, first to peroxide and then to water and oxygen gas. That's the step down. That's the beauty of the system. Where do we see the electron transport chain in the citric acid cycle? We should know that. That's all cell and molecular 101. That's there. So where does it basically occur? Between what membranes do the protons fast? When I didn't even go over it. To me, that's, again, the idea of a prerequisite course like cell and molecular for any of these is you got to know this stuff. So we have concentration gradient. Okay. But what are the other factors that play a role? So in concentration gradient, think of the lung, oxygen and carbon dioxide. If you did not know, oxygen has a much greater gradient going from your lung into your bloodstream in the level of the capillaries and the alveoli than the carbon dioxide, which is a smaller concentration gradient. Yet they move equally. So it's not all concentration gradient, though certainly it's the most important factor. Membrane solubility, size of the molecules are fairly comparable. Okay, membrane solubility is the other factor is there. Effectively, they pass, so we're not really worrying about specific receptors or saturation of receptor size. That doesn't apply so much in simple diffusion, but that's an indicator of that. So when you look at those, that's simple diffusion, and the presumption is nonpolar, non-charged particles. Sodium potassium pumps, what do they do? Okay, effectively, again, this is the distinction between primary and secondary active transport. They're a primary unit. Even though we see particles moving in both directions, that's by design. It's much more a better example of secondary active transport would be as glucose moves in certain systems, it facilitates, I beg your pardon, as we see the proton motive forces, sodium moves in certain systems, and we'll see that in the kidney. Then secondarily, we'll begin to transport in glucose along with it. And we'll see that at the cellular level as well. So some are by design. We will really consider that sodium potassium pump specifically primary, and whether there's or anti-transport associated with it, then those are the secondary aspects of it that are there. Where we see clathrin-coated pits, is it an exocytosis? Is it an endocytosis? What type of molecules generally are associated with it? What role does that play in hyperlipidemias? We talked about that. You know, Again, an absence of certain kinds of receptors associated with those clathrin coated pits, which assist in forming the vesicle. In their absence, it genetically speaking, in the absence of those receptors, that's one of the underlying reasons why we see hereditary lipidemias, which are more common than you would think, and a lot of reason why people have to find other methods to deal with lipidemias and quote unquote high cholesterol, that type of a thing. That's there. I mean, aside, so my family doctor 2 weeks ago said I really didn't care to take that kind of medication, which is called Atorvastatin. Okay, because last time I took it, it gave me a horrible headache, and he basically looked at me and said, "You really don't need it. It's more about diet and exercise with you than it is. It could certainly there's no obvious indicators, nor do you meet the parameters. My numbers were good comparatively. My cholesterol was a little bit up, but my ratio of low density to high density is pretty darn good. It's about 2 to 1. That's not bad. Which means you guys are keeping me active. Okay. You all know about buffering systems. You should all know. You all know about that. Carbonic acid buffer is the biggest player. We're going to hit it so many times in this term, whether we hit it in blood or we hit it in respiratory, or we hit it in kidney, all those kinds of things. Super important. Water and carbon dioxide in the presence of carbonic anhydrase reversibly gives you carbonic acid and then reversibly can break down into bicarbonate ion and protons. It's the biggest player of them all. Proteins and calcium also act as our buffering systems to some extent. So we calcium, even though it only has the valence is 2, it's pretty happy in dealing with buffering aspects. That's why if you chew it Tums, it's made out of it's basically calcium carbonate. Aluminum based one is that's in the old days, there was something called Alka Seltzer, still around, much more popular then. So those were aluminum based. So that's where the Alka came in, alkaline for Alka, and they used an aluminum based agent that was there. Just in case you didn't know that. We talked endlessly about snare proteins and botulism and V snares and T snares. Where are they active? What inhibits their activity, etc. How they work. Dovetail it back in, of course, to the role the voltage gated calcium channels play as we generate either neuromuscular or nerve to nerve function. So, and again, the important roles of sodium extracellularly and potassium intracellularly, balancing with chloride extracellularly and anionic aspects of proteins within the cell intracellularly. What do proteins do in general? We'll go over this again and again, but proteins are a big player, not just because their role intracellularly, because their role in their primary focus that's there. I mean, there's certain properties that are inherent to proteins, one of which is its ability to attract water to it. Again, the only reason it has negative aspects, particularly on its exterior, is it creates a certain amount of polarity and it will have some transient bonding. It's not quite hydrogen bonding, but there's at least a certain amount of attraction that produces the nature of what proteins do in liquid. Typically, they form colloids, depending on how much they are and the nature of the proteins. So, and when we get to the blood, that's super important. That's there. We get into the neuromuscular thing. I have another one of those meat Stein's unique matching. Here are the answers. You have to put the right one with the correct question. Troponin, tropomyosin, G actin, notice the G, calcium, titin. Happy dance. What is what at involved with the sliding filament theory and the sarcomere? Look for all of those. Know the distinction between G actin and F actin, which we mentioned. Calcium, the implication is obviously ionic calcium, good old Ca+2. And remember what titin is, all that kind of elastic stretchy guy. Know about that. Mm-hmm. Now, when it comes to, and again, you have to be very specific. This is, it still applies to you folks. And I mean, I always tell it to the entry level students, the first year students, which I have typically for micro or AP2. The biggest fault that students have is very simple. They don't read questions, period. You don't, oh yeah, I read the question. No, you read the question, but you didn't glean everything from the question. So when I talk about what provokes why isn't this making an action potential? Is it, or I use the term end plate potential, okay? Or is it like in the nervous system, are these just potentials that come at the neuromuscular junction? Are they partial potentials? Do they depolarize or hyperpolarize? There's a difference in that. So if you use the term end plate potential, you know it's a neuromuscular junction. If I use the term action potential, you should know whether or not we're referring to a neuromuscular junction or a nerve to nerve synapse. Okay, all that types of things. Now, in a lot of indications, everything is the same. Okay, voltage gated channels with calcium because it's still a nerve releasing the neurotransmitter. Okay. But when we talk specifically at the motor end plate, what does that eventually do? Once we get the neurotransmitter released, what kind of channels does it strike? Ligand gated channels. Okay. What goes in? Potassium goes in, or does sodium go in? Sodium goes in, potassium goes out. Is that the direct way that we trigger the sliding filament theory? No. That just basically is creating, if it's sufficient to generate an action potential, eventually that will send that potential throughout the individual muscle fiber, and so too around the fibrils by way of the transverse tubules, and so too trigger voltage gated calcium channels intracellularly. So all of that is part of it. So there's a lot of confusion. Read the questions. Am I saying voltage gated or ligand gated? And then where's the location? Okay. Are they in on the nerve? Are they in the muscle? Are they in the second nerve, etc.? The pre or postsynaptic nerve would be the appropriate way to describe it. Understand the sarcomere anatomically. Know what binds the myosin effectively to the Z discs. Know more or less the relationship of the actin filaments to the Z discs. Note the relationship of the active sites on myosin and actin. Remember, it's another confusing issue. Myosin binding sites are on actin molecules. Actin binding sites are on the myosin heads. What's the myosin made of? Those, you know, basically heavy and light chains. How are they arranged? Is there a symmetry to it, to the heads? Are they cocked or are they not cocked? All of the above. That kind of stuff is going in there. Very straightforward. Okay, when we look at the neuromuscular junction, we know it's acetylcholine. We know the role of how it's broken down. It mimics what we're going to talk about on neurotransmitters when you listen to this, or you may or may not have heard that presentation. It involves basically one of three methods, and they're all incorporated in acetylcholine. You get metabolism, what we call degradation, whatever term works. That breaks it down. Acetyl and choline break apart into acetic acid and choline. The choline is reabsorbed, a kind of reuptake, partial in this case. Some of it might diffuse as an entity, and what facilitates the metabolism of it. So there's the three ways: it can diffuse, it can have reuptake, or it can metabolically change and no longer be a neurotransmitter. And in the case of acetylcholine, that is facilitated by the enzyme which we have called acetylcholinesterase. It's not the only place where we have choline esterases. There's a genetic defect in what are called pseudocholinesterases that are relevant in certainly in anesthesia. So if you're heading somewhere where anesthesia is you're going to be your focus, you'll hear more about that. But it's really not necessary for here. So remember what they do. And the flip side is if acetylcholinesterase isn't if it isn't working properly for whatever reason, okay, then the acetylcholine stays there longer. That can be desirable if we're trying to stimulate muscle activity and prolong it. And depending on where the acetylcholine is, because it's involved in different aspects of the nervous system, not just the neuromuscular junction. Now, if we erroneously uh, utilize a true acetylcholinesterase inhibitor, that's powerful. Now we have a biological weapon. We have a big part of chemical weapon. Nerve gas, muscargens, effectively, that's where they work. What they do at the neuromuscular junction is they prolong the activity of acetylcholine to the point where the acetylcholine doesn't disappear, the muscle becomes a spastic paralysis, as opposed to agents that block, like botulism, where it becomes a flaccid paralysis. Tetanus is slightly different because of its mechanism. Its toxin works more appropriately not at the neuromuscular junction, but at the junction between the, the upper motor neuron and the lower motor neuron. So that's where that generates its problem. It blocks the neurotransmitters that induce relaxation of the muscle. So every stimulus to the muscle, and they're always being stimulated. We always have muscle tone. Effectively, is excitable or depolarizing rather than non-depolarizing. So I don't know if I mentioned that little nugget about that, but that's when you hear about sarin gas or VX gas or mustard gas. That's how they work. DDT. We used to do that as an insecticide. That's how it works. And it's why, if you probably were, there's a lot of it. You've got to be more careful today. There's been a lot of lawsuits with regard to certain insecticides that were used over time, even though they were very efficient. But the trouble because people suffered when they came into too much contact with them. The A and I bands are also part of this. There's a lot of material. So what's the A band? It's about overlapping. The I band's where it doesn't overlap. The changes are not so much in the length of the A band, but its appearance, because basically it loses that middle or H zone as the fibers. And really that's the limitation. Once all the actin filaments are pulled as close as they can to that middle zone, now it all appears to disappear. Now we have one unified A band and a much shorter I band. Again, muscles rely specifically, and skeletal muscle, this of course, rely on glycogen. Okay. Only glycogen will do. As a typical emergency energy, will convert it and things like that. Obviously, the energy is useful, but the stored energy within the muscle is glycogen. The stored oxygen, myoglobin, which is released a little more quickly than hemoglobin. We'll look a little bit at myoglobin and hemoglobin curves sometimes with some of the articles or some of the discussions we have. Then the whole nugget of what we do from a rehab or diagnostic sense with regard to stimuli. And so the amplitude of the signal is a lot less about how powerful the stimulus was rather than, again, how frequent. You remember the graph? When we stimulate and let it completely relax, we get a twitch. So if we do one of those every 100 milliseconds or 10 per second, it'll just be an isolated switch. We can do it in the laboratory. We can then see how the muscle is responding. We can look at the latency period and things along those lines. And we can do that by let's say, going to 20 of those a second. Now we're going to get unfused. We're going to get staircasing. And the staircasing shows how additional units are being recruited because we're not giving it enough time to rest for the intracellular calcium to go back into the terminal cisternae, and voila, we have another condition. Now we'll jump it up to 30 a second. Okay, now we're going to get unfused tetany. It's going to quiver. Okay, and we do that. We're trying to achieve certain things also to see if the muscle's responding appropriately. Now the infinitesimal amounts, tiny amounts, let's say, of calcium are being reabsorbed. That's why we get that quivering. And if we go to like 50 a second, now we get fused tetany. It just goes right and goes into a spasm. Why is it desirable? Because we can, in the lab or in physical therapy, we can induce spasm, prolong it. Our goal to fatigue the muscle so it will finally relax and the spasm will break. Breaking spasm is not that easy. And we have a lot of experiences with back spasms, and, trying to use this technique rather than bombard somebody with medicines. So you broke spasm, but they couldn't function anyway because they were so doped up. for lack of a better way to express it. So all of that. So when we get down to it, whether it's a nerve or a muscle, the key to the action potential once you reach threshold is first voltage gated sodium and then voltage gated potassium channels. And yeah, the inactivation gate plays a role. We don't see that in the muscle fiber, but we see that. We see voltage gated sodium and voltage gated potassium because those sequential channels will cause that wave of action potential, that wave of rapid depolarization to repolarization occur throughout the fiber or the fibril, as it were, in the individual fibrils, as well as the fiber itself in all directions. And again, don't forget the 3-2 ratio. That's super important. 3 sodiums in, 2 sodiums out. We reverse it with the sodium potassium pumps. 3 potassiums out, 2 sodiums in. They're constantly working. They just work very, very hard when we're using those particular nerves or those particular muscles. The terminal cisternae are certainly part of the sarcoplasmic reticulum. The T tubules are part of the sarcolemma. But together, that area where the T tubule juncts with terminal cisternae along the AI band junction point on either side is called a triad. And that's where the voltage gated sensors are located, both in the T tubule and the terminal cisternae to facilitate the outflow of calcium intracellularly to trigger the sliding filament reaction. So all of that. So the nerve impulse is obviously going to be in the sarcolemma. Sarcoplasmic reticulum is a structure, in this case, as many of them do, smooth endoplasmic reticulum that becomes wells for calcium and other products. Nothing unusual about that. So you'll be able to see all that. We reviewed the changes in the A and I band, etc., etc. We know that as long as there's calcium there, it's going to keep going. Okay. So again, when we add calcium to the party, it will continue to happen. We'll have greater contractions. They'll be more frequent. Yes, eventually they'll be limited, but not until it reaches tetany. Okay. And then the energy utilization, remember that effectively it's anaerobic to start. We don't convert to aerobic until at the very least we start to use the muscle. We used the initial storage of materials that were in there, the available ATP, and the available sources of oxygen that were present, whether in the, as I said, in the glycogen or in the myoglobin. After that, creatine phosphate. And we utilize that high active creatinine phosphate creatine related nugget to use for additional. First one's 4 to 6 seconds, second one's maybe another 10 seconds. Then it's full blown anaerobic glycolysis. Okay. And the eventual waste product, eventually lactic acid. We liberate aerobic, liberate CO2 and peroxide. And it's not, and the lactic acid is not the only quote unquote, that's the principal, let's call it organic waste product. There's other materials that are produced. Let's say it's the accumulation of electrolytes and other materials, but I don't think they're technically classified as waste products when we look at the organic. When you look at either lactic acid, carbon dioxide, and water for aerobic, lactic acid for anaerobic. And again, it functions differently because not so much the lactic acid, which we can utilize and we can revert back to pyruvic acid in the liver and make more glycogen, etc. It's the other materials that cause spasms and impair function as well. And that's very interesting. We get into studying the actual things which they do for muscles or they do now in sophisticated physical therapy and athletic training. Okay. And so again, just to reinforce the lactic acid, your liver takes care of that. That's the old, what they call the epoch. I just call it an oxygen debt, back from where we came from. That's an interesting chart about you have to be in that golden zone of 80 to 120. Obviously, optimally would be 100, but if you went as low as 80 or as high as 120, you still have enough contact points along the thin and thick filaments to satisfactorily generate power and movement in those muscles. It's neither overly contracted nor underly contracted. Okay. And truly, motor proteins change shape. So if it's not moving, if it's not changing its configuration, it's not a motor protein. That are there, uh, different things. So if you're thinking of different kinds of activity that are there. So look at the roles where proteins are. Myosin is pretty much exclusively in muscle activity. Actin's a little bit different. So actin plays a pivotal role in muscle contraction. Actin plays a pivotal role in mitosis. It's tiny little actin filaments that that start to create motion, particularly if you want. And when we talk about mitosis, I'll make the inclusion of cytokinesis because that really makes the cleavage furrow. That's part of it. That's there. So I mean, I will make an adjustment in a question to clarify it because I don't want to. I hate ambiguous questions. And certainly, amoeboid motion of macrophages. That scooching, that movement of cytoplasm, is facilitated by that. You understand? And after the test, I'll show you the difference, how you can upgrade the levels of questions as well. But I'll make a mental note to make that change. And the question, when we get to class, I'll add it appropriately. And let's see. Okay, where this is asking about where the location is of voltage gated channels are there. And so do remember where, when you're talking about nerve or muscle, where those channels are located. They're principally in the membrane, whether it's the membrane of the nerve or the membrane of that we now call the sarcolemma and the sarcomere. Okay, the role of glial cells I talked about at length. We just had that. So what do they make? They make myelin. They organize. They support growth and development, organization, a certain amount of protection, nutrition. They do basically everything. It's almost nothing they don't do with regard to those aspects. Do they make neurotransmitters? No. Okay. Understand that. Do they help? Do those ependymal cells play a role in cerebrospinal fluid and how it moves around? Yes. Okay. Myelin, yes. Okay. Whether it's oligodendrocytes or if we go in the other direction with Schwann cells, do they have uh, immunological activity or defensive activity? Microglia certainly have that. So there's lots of things that they do. So know what the different names of the glial cells and watch the questions, whether it says central or peripheral nervous system, because you have different guys. The dog is changing the channel. It looks like he wants to see a full Bee Gees concert from 1997 on YouTube. Well, I didn't know you wanted to listen to that. I'll play that for him. Don't forget the blood brain barrier. Again, it's all about the nature of the capillaries and it's reinforced by the astrocytes. Capillaries are unique in that area. They're continuous capillaries, which we looked at when we looked at the anatomy of the bloodstream, but the little clefts aren't really functional because there's so many tight junctions. That's what prevents the water soluble base movement. And then it's surrounded by glial cells both on the capillary side and on the nerve side, so they have to traverse through the glial cell. So it's a combination of things, but the capillaries with a tight junction and the covering of glial cells is the big player that's there. So again, CNS versus PNS, CNS oligodendrocyte. Remember, and the myelin is different as I'm talking about the myelinization. The oligodendrocyte's different, multiple. So oligo means a few or more than just a little number of nerves are involved. And yet the way that they wrap around several nerves at a time, and then the spaces are between oligodendrocytes, as opposed to one long axon with Schwann cells wrapped around the one long axon and then separated from each other, and then you have the nodes or the sheath caps. Again, smooth and thick, different. The myosin to the actin to myosin ratio is about 2 or 3 to 1 in skeletal muscle, about 13 to 1 in smooth muscle. That's why it appears smooth. You just don't have enough filaments to create the overlap that is there. We're getting close to the end. You'll be happy to know. You certainly should know about the distinction in smooth muscle. Calcium is mostly extracellular, though there's some intracellular. It bonds to calmodulin. There's no troponins or tropomyosin. Yes, there's myosin and actin in the sense that they're more along and they're held together by those dense bodies. Okay, it's all involved. The calmodulin chain and shape activates the myosin kinase that facilitates that sort of unique spiraling contracture that's there, and so it really is significant. And again, the dense bodies are kind of like the Z disc. They're sort of a proteinaceous structure. Okay. And understand skeletal muscle is powerful, smooth muscle is not. Smooth muscle is all about endurance. Okay. And really, when you talk about smooth muscle, it's about extracellular calcium. When you talk about skeletal muscle, it's intracellular calcium. And when you get into more sophisticated questioning, depending on where you're going, again, look at where the source of the calcium is and the uniqueness of the contraction that's there. Don't forget the role and the particular role of the inactivation gates and what they do, because I made a big point out of it, with particularly with absolute refractory periods and some overlap into relative refractory periods. I have to rethink this question. I'm gonna have to reword it. I don't. I think when I made one of these questions, I'm a previous author, so I might have to throw it out. So let me skip it and see if I can edit it. And if I do, then I'll comment on it in class. So hold on. Okay. Again, the beauty of the nervous system, more so than the muscular system. The muscular system works very rapidly for skeletal muscle, slower for smooth, slower yet, even though it's a quarter of a second at absolute refractory period. But the big player when it comes to nerves is it's really a tiny absolute refractory period. Okay. And a lot of that has to do with, perhaps more than anything else, is that there's just so many sodium potassium pumps that allow that to occur. They're able to reset that very, very quickly. Number one on the hit parade. Okay. It has, so really, it, it's typically a, a very significant issue that will reset the cell and readjust it. And the other factor that plays a role are voltage gated potassium channels that play a role. So again, it's another question I'm going to have to reconfigure. I'm seeing this now as I review the test that I have made some elements that are part of it. So I'll clarify that as well. Again, stimulus intensity is related to frequency. Very straightforward. There's nothing really you have to say about that. The action potentials are identical. That's the all or none principle. Again, what makes a nerve less likely to fire? If it becomes hyperpolarized. Think of what makes it hyperpolarized. Okay. What's going to make the inside of the cell more negative? Okay. Think of those things. Okay. So, what's going to make a cell more negative on the inside if it loses potassium? That'll make it more negative. If, and that's the start of the resting membrane potential. Sodium entering makes it more positive. Chloride or negative leaving it would make it more negative. So understand those are the things that are part of it. Sodium leaving it, but it really doesn't do that other than by the pump mechanisms, also theoretically would make it more negative. And understand that specifically end plate potentials are only seen in the neuromuscular junction. Okay? That's the significant factor that are there. So when we talk about axodendritic without anything, that synapse between axon and between axon and dendrite is presumed to be in the nervous system. That is there. Let's see. So, lastly, we have a sequence question. You'll hate it. It comes at the very end. Always put hard stuff at the end, bothers students. Really, because there's a fatigue factor. What happens? Okay. When a nerve basically tells another nerve to fire. Voltage gated calcium channels cause the exocytosis of whatever the neurotransmitter is. The neurotransmitter, what does it do? If it's sufficient, and it's not just a localized potential or a graded potential, if it's sufficient to meet threshold, an action potential occurs. What happens then? The action potential will reach out to voltage gated sodium channels that trigger depolarization. They are followed by voltage gated potassium channels. But in between that, to make sure that the sodium channels no longer impacted, they become the voltage gated sodium channels inactivate. Then the voltage gated potassium channels open, okay, and achieve repolarization. Only then do sodium potassium pumps activate. Be familiar with all those events. Voltage gated, and this is nerve to nerve, voltage gated calcium channels, exocytosis of neurotransmitters, graded potentials or limited potentials, or any way you want to look at them, not end plate potentials, no end plate apply. They generate an action potential. If it's sufficient to be threshold, it's an action potential. First stop, voltage gated sodium channels. Then once they're done, they inactivate. Inactivation gates swing into the area. Then the voltage gated potassium channels. Then they repolarize. And at that point, that's when the sodium potassium pumps reset the internal. Again, in this case, those molecular events are going to have to be reset. And when we have to reset the molecular conditions that are there at rest, and that's what the sodium potassium gates do. And that's everything for now. I'm going to make a few little tweaks, and I'm happy I did it this way so I could run over and see where the problems were in the test. So I'm going to tweak a couple of questions. I may hand them out separately. I think that's the way to do it. All right. So I may hand out a couple of pieces of paper with, strike the one question and change it to the language, because I've already got the print shop working on it. Thanks again, and see you tomorrow.